I'm going to ask our wonderful and excellent presenter, Dr. Zaira Vergara, to join me on the platform. And she's going to teach us about Citrus, really a powerful tool that she taught me so much about. Prior to this, I was really in clouds trying to understand this, but Zaira is going to teach you a lot about it.
Almost 85% of think about the data analysis prior to setting up studies, and that's great because, as you may know, sometimes if you don't do that, then you end up at the analysis time and you realize, oh, I need an extra control, or I didn't acquire enough samples of this group that I want to compare, etc. So that's great. And I'm really happy to learn about that. So with no further ado, let's look at today's agenda. And today we are going to be talking a lot about Citrus. So the first thing that we have to do is to define what exactly is Citrus. Then we will see what do we need in order to run this analysis. Then we will follow to visualize how the analysis works. And with all that information, we will move into a case study that we are going to analyze together. So I hope you are engaged at that moment and you have fun, as I did, analyzing the data. And finally, we will see what else can we do with our Citrus findings. So Citrus, apart of being a group of fruits, it's a data analysis tool that was first published in 2014. And it combines two very important analyses, a clustering analysis and a statistical association analysis. And like that, Citrus is going to generate different models that are going to identify in a predictive or in a correlative way the differences between groups of samples. And the reason why this is going to be very interesting for us is because with Citrus, we are going to be able to give an answer to very biological relevant questions. For instance, maybe on your clinical research, you are looking at individuals that respond to immunotherapy versus individuals that don't. Or very important nowadays, maybe you are asking, why does a vaccine elicit immune memory in some and not in others? Or just, why do some people recover quicker from surgery? And in any case, for all the different clinical research questions that you may have, Citrus is going to give us, biomarkers. Yeah, Citrus is going to give us biomarkers that can help us predict outcome ahead of time. So by now, I guess all of you are willing to give it a try and run a Citrus analysis and are asking, what do I need in order to run it? And a good start is to define at least two groups of 8 samples that you want to compare. And then we will need to make some decisions because there are several Citrus pathways that we can follow, and we are going to choose one or another depending on our biological question, the panel, meaning the list of parameters that we have analyzed, and our need or not to make predictions. So first, we need to ask a biological question. Am I looking for differences in the frequency of cells within a specific cell type, or am I more interested on the expression level of some markers? No? So for instance, am I looking for, is the frequency of B cells important to create protective immunity after vaccination, or on the other hand, are the cytokine responses implicated? And of course, we are going to choose one pathway or another depending on our panel. Typically, if we only have information about the lineage markers, we are going to look for differences in the abundance of cells, while if we have information about functional markers, those cytokines that we were talking about, maybe we want to focus on how those cytokines are expressed, looking at the median signal intensity. In any case, we are going to have the option to run Citrus in the correlation mode or in the prediction mode. With the correlative tools, we are going to identify all the significant clusters between the groups that we are comparing, while the predictive tools are going to identify only the features that are needed to predict the outcome. So by now, we have our groups of samples that we want to compare. We have think about what are we asking, and we have choose the Citrus pathway that we have to follow. And then we have to see how the analysis works. So here we have some lymphocytes from group 1 and group 2. Now video is loading on your website. And the first step that Citrus is going to do is what is called the preparation of the data. And that means that Citrus is going to combine all the cells together in an initial clusters where cells from group 1 and group 2 are going to be mixed together. After this initial cluster is done, then Citrus is going to start the clustering analysis, creating an agglomerative hierarchical cluster. That means that it's going to separate this initial cluster into smaller groups of cells, always focusing on phenotypically similar cells as the green and blue lymphocytes that are already separated. And it's going to create smaller and smaller clusters until it reaches a minimum cluster size. Because this is an agglomerative hierarchical clustering, we are going to have mother, child, and grandchild clusters, and some cells are going to be present in more than one cluster, as you can see there for the orange lymphocytes. After the clusterization is done, we have already our citrus tree, but all the cells are mixed together. So what Citrus is going to do is divide the cells back to the original sample and the original groups. So now everything is mixed together, and now Citrus separates the lymphocytes into the ones belonging to the group 1 and group 2. After this separation is done, Citrus is going to describe all the clusters in a per sample manner. That means that it's going to annotate in a table all the different features from the clusters, like the abundance of cells, or the median and signal intensity, depending on what we have chosen. With that information, Citrus goes through the statistical modeling, and like that is going to state which clusters are statistically significant. So right now we have visualized how the clustering step works for Citrus. And with that information, what Citrus is going to create are models. And those models are going to associate biological signatures with the defined groups, with our defined groups. If we choose to choose the correlative mode, or we are going to see the correlative models, we will use the algorithm SUM. And SUM is going to identify all the significant features between our groups at 3 different fold discovery rates that are 1%, 5%, and 10%. On the other hand, if we use the predictive mode, if we look at the predictive models, as we will do today, we are going to have 2 algorithms, either PAM or LASSO. But both of them are going to identify the features that are needed to predict the outcome in new samples. And both of them are going to use regularization and cross-validation to show us the model that better predicts the outcome. With the regularization, we are going to prioritize the simplest model. And with the cross-validation, we are going to obtain an error rate, a measure about how many times our models are correct and how many times our models are wrong. So like that, the model is going to be trained with part of our data, and then it's going to be tested with the rest. So now that we understand how Citrus is working on the inside, let's go to our case study. So when we started with this case study, we set up different goals. First of all, we wanted to identify the biological signatures that were statistically different between groups of individuals. And at this point, I have to make a note, because the parameter to segregate the samples in the 2 groups was unknown. Our second goal was to characterize the statistical significant cluster, understand what are those clusters and what is the difference between them. And our third goal, and I hope you help me with this one, is to decipher what might be this differentiating factor between the 2 groups of individuals. So please stay tuned, stay alert, and at the end of the case study, we will have another pool. So our experimental method started by collecting blood samples from 16 healthy donors. Those samples were stained with antibodies against phenotypic markers, and the samples were analyzed by flow cytometry in a Cytoflex Select to ensure maximum standardization at the acquisition process. After that, the data was analyzed in the Cytoflex platform, and we used Citrus in order to compare our 2 groups of individuals. At the staining step, to mark the samples with the phenotypic markers, we used the Duraclone tubes. Duraclone tubes are Beckman proprietary dry reagents. They are ready to use and a perfect way to obtain more reproducible results. We use the Duraclone IM T-cell kit with some drop-in antibodies in order to create an 18-color phenotypic panel. After the samples were stained on the Duraclone tubes, we acquired the data on the Cytoflex Select, and then we pre-process the data on the Cytoflex platform, meaning that we ensure good compensation of our cells and a good scale transformation, and we pre-gate it on the lymphocytes as our population of interest for today. So let's go to the Cytoban platform and run one citrus analysis together. And you will have a second video that is going to show in your screens in a second. So, okay, it is loading now. So once we are on our experiment on the Cytoban platform, the predictive power of citrus, we have done other analysis here, but today we want to run a new citrus analysis. So we have to select that, advanced analysis, new citrus, and we have to give it a name. So for instance, citrus lymphocytes. The first step would be to select the population of interest. And as I said, we are going to use the lymphocytes. So here I have a lot of manual gates that I have done, but let's use the lymphocytes. If you're creating your experiments, this analysis on your experiments, feel free to use other populations. Then we have to select the different groups. We have to create the groups that we want to compare. So for that, I'm going to add the different groups, and I'm going to call them group 1 and group 2, separated by commas. You can add more than 2 groups. You can compare more than 2 groups, but at least 2, of course. And my samples have been automatically assigned to the correct group because I do have the name of the groups on my file samples. So Cytobank understands that and automatically assign correctly my samples. Otherwise, you can do that manually. And we also have information about how many cells do we have in every sample and how many cells do we have under the lymphocyte gate. The third step is to select the channels, the parameters that we want to use for the clustering step. And I'm going to use all the different parameters that I have, except forward and side scatter. That's not very interesting. Not the heights, of course. I want only to use the areas. And I don't need the viability and the CD45 markers because I use those to gauge my lymphocytes. So let me check that everything is correct here. And yeah, we can see 16 colors. Then we have to select which association models we want to use, and we have 3 options here. We can select the 3 of them, so we can choose correlative and predictive if we want to. SAM, remember, is the correlative model, PAM for prediction, as well as LASSO for prediction. LASSO would be only available if you have 2 groups, as we have today. Then we have to choose between the abundance or the medians, what we want to compare. No? Today we are going to use abundance. We are going to look for differences in the frequency of cells. But you can also use the median mode if you have a different panel with more functional markers, etc. So if you don't want to use the abundance, you want to use the median, then you have to select which channels you want to use for the median signal intensity comparison. And just make sure that typically the channels that you use as statistical channels are not going to be the same as the one that you have on the clustering channel. So today we are going to use abundance. And the next step is the event sampling. That can be either equal or maximum per file. Maximum per file is only available for median, so we are going to use the equal event sampling. And finally, we have these advanced settings, these additional settings that we have to also take care of. First, we have to select how many lymphocytes, how many cells per sample we want to include, and we will have information about the total number of cells that are going to be included. Then we have the minimum cluster size. So remember that the clustering is going to create smaller and smaller clusters until it reaches this minimum cluster size. And if you can use it like a magnifying glass to see the small populations of cells. And because I want to characterize really well the data here today, I'm going to use a 1%. So like that, we will have smaller clusters. Then we have the cross-validation folds. And this is the number of groups that we can separate the samples for cross-validation purposes. The higher the value, the more robust the test. But 5, the default number, works very well, and that's the one that I'm going to use today. Then we have the false discovery rate. We can be more restrictive with our analysis by adjusting this percentage to something lower or more permissive and increasing this percentage. And today I will also use the default 1%. Then we have the option to further transform our data by normalizing the scales, meaning apply a z-score to all the parameters. And finally, we have the option to choose between white or black background for our results. Once we are happy with our settings, we can click on Run Citrus Analysis, and now the order is on the cloud. The cloud is going to be the one that is going to run the analysis, not on our local computers. And once everything is finished, we will get a lot of different information. We are going to get a lot of PDFs with all the models and all the information about the models. So today, one of our purposes, it was to test the predictive power of Citrus. And for that, we are going to focus only on the predictive results and not on the correlative ones. Of course, when I make a prediction, I can be correct, I can make a good prediction, and I can make a very bad prediction, and I can be totally wrong. So there is a way to check if our models are going to have, we can check the validity of our models to ensure good prediction. And for that, Citrus is going to provide us with the model error rate plot, like this one for PAM. So the first step when you are running Citrus in the predictive mode is to go and have a look at the model error rate plot. Because if it says that your prediction is not going to be good enough, then go back to your settings, but don't go and explore the models. And here we are going to have a lot of information. We are going to have 2 different rates and several models. So in red, the line that you can see in red is the cross-validation error rate. And this is going to tell us the percentage of time that the model is wrong at predicting the groups. So as you can see on the left of the graph, the cross-validation error rate is around 50%. That means that for those models, it is the same as tossing a coin, because 50% of the times the models are going to be wrong and 50% of the times the models are going to be correct. But luckily for us, our cross-validation error rate decreases and even reaches 0. So there are some models that are very good at predicting. Then we have the false discovery rate in blue. And this rate is going to tell us the probability to get false positive features. And of course, it increases with the number of features. So you can see how the blue line increases on the right part of the graph. Apart from having these 2 rates, PAM is going to generate 3 different models that are the minimum, the one standard error, and the false discovery rate constraint. So as you can see, the little dot in green, that's the minimum model, and that's the one with the lowest cross-validation error rate. In orange, you can see the one standard error, which is the model with the cross-validation error rate that is equal to one standard error higher than the minimum. And this is a factor using statistics. And finally, we have the yellow model, which is the false discovery rate constraint. And this is the one that contains all the features below the false discovery rate that we set up. And as you remember, we use it 1% by default. So here we have our model error rate plot for PAM with the 2 rates and the 3 models. And evaluating that, we can say that our models for PAM are going to be quite good. But if you remember, there are 2 algorithms for the predictive mode, PAM and Lasso. So let's have a look at the model error rate for Lasso. And Lasso does not use the false discovery rate, and that's the reason why we don't have the blue line. We only have the cross-validation error rate. And as it happened for PAM, there are some models that contain a cross-validation error rate that around 50%, but then it decreases and it goes to 0, which is very good. Apart from that, Lasso is going to generate 2 different models, the minimum that you can see in green and the one standard error that you can see in orange. Because Lasso doesn't use the false discovery rate, we don't get the false discovery rate constraint model. So, okay, these 2 model error rates look very, very good, but unfortunately, sometimes we are going to obtain more unreliable model error rate plots. And I want to show you some as an example. So here we have 3 examples. The first one on the left is not very bad. So if we look at the cross-validation error rate, it is true that it doesn't go to 0, but it goes below around 10 or 5%, which is an error that maybe you can accept. And the false discovery rate doesn't go very high until the end, so that's good. And we have our 3 models for PAM. Now, the model error rate in the middle, that one does not look very good. First of all, yeah, we can see that the cross-validation error rate goes down again to 5–10%, so not too bad. But the false discovery rate goes high very from the beginning, and we don't even have the false discovery rate constraint model. And the one on the right, well, this one looks worse. The cross-validation error rate doesn't go too low. Well, it goes to 30%, which could be acceptable in some cases, but the false discovery rate constraint is very high from the beginning, and we also lack the false discovery rate constraint model. I would like to tell you that these 3 examples of model error rate plots were obtained with the same data set that we are analyzing today. So please, if you run your citrus analysis and you end up with something like that, don't be discouraged. Just let us know, and we will be happy to help you. And maybe there is something that we can analyze together with your data. So going back to our case study, remember that we evaluated the PAM and the LASSO model error rate plots, and they look very good. So now let's assess the models. And I know that we have 5 models in total, so 3 for PAM and 2 for LASSO, but we are going to look only at the minimum models for PAM and LASSO. And here we have them. So we have the citrus tree with all the different clusters. And now we have a lot of clusters, not like in the video, that was a visualization of what was happening over there. And the clusters that you can see in orange are the ones that are identified as significantly different clusters suitable for prediction. And as you may notice, the ones in PAM and the ones in LASSO are, except one, they are different. So which clusters could be more useful for us? Today, remember that we have set up different goals, and one of them is to decipher the stratifying factor between our group 1 and group 2 of individuals. So one way, like we can have more information, is to inspect the citrus tree with the marker expression levels. So here we have our citrus tree, but now it's colored by the level of expression of CD3, from blooming in a little to reddening in a lot. And like that, we can annotate in which part of the citrus tree the T lymphocytes, the CD3 positive cells, are going to be located. We can do the same for our B cells, looking at the CD19 expression, and we can mark that also on our tree. We can also do that for the CD56, for the NK cells here, but we can go one step further. And for instance, we can look within the CD3 T cells, which ones are expressing CD8, and which ones are expressing CD4. And we can even go one step further and look into very specific details. So for instance, we can look within the CD4 positive cells. We can spot one cluster that is highly expressing CD25, and it is also lacking CD127. So now that we have inspected our citrus tree by looking at the marker expression on the tree, we have realized that the more specific clusters of cells are located at the end of the branches of our citrus tree. So going back to our models from PAM and LASSO, it is true that we identify more clusters with PAM. We identify 9, but they are at a higher level lineage differentiation, while the ones from LASSO are at a deeper level of cluster segregation. And today, we really need to separate the specifics, because we don't know what is different between group 1 and group 2, and we really want to decipher that. So let's focus on the LASSO association model. So here we have it, and we can give names to those 6 clusters from A to F. Citrus, a part of creating this citrus tree, is going to provide us with histograms and box plots to understand what are those clusters that are labeled as statistically significant. So let's have a look at first the clusters A, B, and C, because they are located in one part of the citrus tree, as we saw before. And here we have our histograms for the different clusters and the box plots. If we focus first on the histograms, we can see 2 different lines. The blue one is the background, meaning all the cells that we have in our analysis. And in orange, we have the cluster cells. And like that, we can see that clusters A, B, and C are going to be CD8 positive cells. And they are going to be naive CD8 positive cells because they are also highly expressing 45RA, CCR7, and CD27. And the main difference between clusters A, B, and C is the level of expression of PD-1. In any case, for these three clusters that are CD8 naive T cells, if we look at the different box plots, we can see the difference in abundance between group 1 and group 2. So in group 2, we have it labeled in light blue, and in orange, group 1. And in any case, we have more frequency of those cells, of these CD8 naive T cells on group 1. So we have identified the first biological signatures that is different between group 1 and group 2, which is that the CD8 naive T cells are more abundant in the group 1 compared to the group 2. Now let's have a look at our other three clusters, clusters D, E, and F. And again, we are going to have our histograms and the box plots. So let's have a look at the histograms. And again, we have the blue line for the background, meaning all the cells except the ones in the clusters, and in orange, the cluster cells. And we can see that in this case, the three clusters are CD4 positive cells. And for the cluster E, we can see that those are going to be Tregs because they are highly expressing CD25 and they lack CD127. And when we look in the box plot about the difference in the abundance of those Tregs between group 1 and group 2, we saw that those cells are more frequently present in group 2. So our second biological signature is that the CD4 positive Tregs are more abundant in group 2 compared to group 1. So as a result, after going through our citrus analysis, we have found that the two differentiating phenotypes between group 1 and group 2. First is the abundance of naive CD8 positive T cells. And we have seen that there are more naive cells in group 1. The second one is the abundance of Tregs. And we have seen that there are, Tregs are more abundant in group 2. So now that we have two biological signatures that define these groups, and here we have our original data, so how the data was stratified, and we have here our group 1 with the 8 samples and group 2 with the other 8 samples, and we have an information about the age. And we can see that group 1 are younger compared to group 2, and group 1, remember, they have more naive cells and less Tregs. So now that we have analyzed our case study, what else can we do with those citrus findings? And something that you can do is, going back to the Cytoman platform, we can export those clusters that are very important for us into a new experiment. And for that, we have to click here on this option that says export the clusters. But of course, before we export the clusters, we have to identify the real cluster ID. So here we were talking about clusters A, B, C, D, E, and F, but unfortunately it's not that easy. This is how the citrus tree really looks like. Every cluster is going to have an ID that is going to be for that particular cluster only. In here we have very big. Our numbers, don't worry, you don't have to write it down. It's just something that you need to know because when you export the clusters, you're going to have the entire list and you have to look for the correct IDs of the clusters that you want to export. Here we have our 6 clusters. You can also include the original files if you want to. And then you just have to click on Start Export. And this is going to generate a new experiment with independent FCS files for each of the clusters in a per sample manner. With that information, we can create new illustrations, create new reports of statistics, and very interestingly, and this is what we are going to do today, we can combine our citrus results with other algorithms that are on the Cytoman platform. Today, we are going to combine citrus with VISNE to better visualize our findings. And VISNE is a dimensionality reduction algorithm that is going to help us visualize this high-dimensional data in a better way. Like that, the cells are going to be displayed in a two-dimensional plot according to the marker expression for all the different parameters. So what we did is, first of all, of course, export the clusters and the original samples. We wanted to have like this route to the original samples. Then we ran some business analysis, we concatenated our files, and we compared them. Because remember that we have used the same number of cells per sample in our citrus analysis, and we also have the same number of samples per group. After running the citrus analysis, the business analysis, this is an example of how the complete sample looks like. Not just the cluster, but all the different events. And we can color our business map with different markers, and here we have it colored by CD8. So we can see in red the cells that are CD8 positive. We can do that not only for CD8, but for other markers. So here we have on the left our group 1, the total sample, and we can see the regions where CD8, CD45RA, and CCR7 are expressed, and the same on group 2. Of course, when we are looking at the entire sample, it's not easy to spot the differences, no? But if we only look at cluster A, for instance, we can already see that there are more cells, there are more dots on group 1 compared to group 2. And because we use the same number of cells per file and we use the same number of samples per group, we can even compare the event count. And here on the right, we have the box plot that gave us citrus. And again, we can confirm that these cells are more abundant in group 1, which were the younger. We can do the same with other markers. Now, not to spot the CD8 positive cells, but the CD4 positive cells. And here we have also information about the CD25 and CD127. And of course, now we are going to look at our cluster E. And here we can see that there are more cells in group 2 compared to group 1. And again, because we use the same number of cells, same number of samples per group, we can compare the raw number of cells, and we can see that there are almost 3 times more cells in group 2 compared to group 1, and those were our Tregs. So just to summarize, today we have been using the citrus data analysis that enables us to identify predictive. Today we have focused on predictive tools, but it also gives us information about the correlative differences between groups of samples. And like that is going to help us characterize different groups of samples in a meaningful biological way. In our case study, we took different healthy normal individuals, we grouped them by age, and after running a citrus analysis, we find out that 2 biological signatures that define these groups is that there are more naive CD8 positive T cells in younger than in older, and that there are more CD4 positive Tregs in the older. The power of citrus can help you identify predictive biomarkers that can potentially predict an outcome of a therapy or help you identify the therapy with the better outcome. And most importantly, the power of citrus is accessible to you without the need of coding. And thank you all for your attention. I would be very happy to answer any questions. And please stay tuned for our next Cytobank webinar.